Hello, prospective graduate students. My name is Arun Gupta. I'm with the Department of Chemistry and Biochemistry, where I'm a professor. I'm also affiliated with the Department of Chemical and Biological Engineering at the University of Alabama. I'm going to provide you a brief overview of the research activities in my lab in the next few slides. So let me give you overall perspective. My work primarily involves material synthesis, assembly, and applications. A variety of applications, including spintronics, solar cells, biotechnology, and sensors. And these materials are primarily inorganic-based materials. We do controlled synthesis and assembly of thin films, nanomaterials, and nanostructures, emphasis on the exploration and manipulation of their physical and chemical properties. We implement a multidisciplinary approach, which includes chemistry, material science, physics, and biology. And the inorganic materials we investigate are different types, including metals, oxides, chalcogenides, which exhibit diverse optical, electrical, magnetic, and semiconducting properties. Let me give you a snapshot of the three focus areas first. First one involves thin films and heterostructures for spintronics. We grow controlled heterostructures of inorganic materials in the form of thin films, as shown here for two magnetic layers, complex oxide magnetic layers, separated by a very thin insulating layer. Note the scale here, this is 2 nanometers. So these two magnetic layers are precisely separated by 20 to 30 angstroms. Similarly, on the right-hand side, I show you the University of Alabama logo, which has been written on a ferroelectric oxide material using atomic force microscopy. And again, note the scale here, it's 1 micron. So this can be stored indefinitely in this form of ferroelectric material. The second focus area is on nanoparticles and films for energy and biomedical applications. What we do here is to actually grow precisely controlled monodisperse magnetic oxides, for example, or semiconducting chalcogenides, where we can control their properties by their shape, size, or by the composition. So we can change the band gap of the material, which leads to different colors of the solution by changing the composition of indium to gallium in this complex chalcogenide, which is indium gallium-based copper chalcogenide. Finally, an important focus area is on nanobio assembly. What we do here is to use viruses, bacteriophage viruses, which are harmless to human beings. We engineer them either on the surface, top surface, or on the inside, precisely grow three-dimensional inorganic architectures. So let me dive into each of these three focus areas and give you a flavor of the kind of work we do in them. The first one involves spintronics. Spintronics refer to spin electronics. You know of electronic devices where we primarily use the charge of the electron and we transfer the charge from a transistor, from a source to a drain by modulating the voltage on a gate. So we can actually make different devices for different applications. What we want to do here is, in addition to charge, use the spin of the electron, which can be spin up or spin down, to add additional functionality to devices. So such devices already exist. They include the giant magnetic resistance, or what's referred to as GMR, or tunneling magnetic resistance, which are EMR, and they are used in devices including in the hard drives. An active field of research is spin transistors, where you actually make similar to electronic transistors. In addition to the transport of charge, we manipulate the transport of spin and thereby have added functionality. The ultimate goal is to use these devices in the emerging area of quantum computation. One such type of device are what I refer to as tunnel junctions, where we have two ferromagnetic electrodes similar to what I showed you in the previous slide, separated by a very thin insulating layer. By changing the magnetization of one of these ferromagnetic electrodes, we can change the resistance of this device and hence make different memory states. Instead of using ferromagnetic layers, we can also use non-magnetic metals for the electrodes and separate them by a ferroelectric layer. And by changing the polarization of this ferroelectric layer, again, we can store information, memory information in this type of device. And finally, by using a combination of ferromagnetic and ferroelectric layers, we can make multiple state memory devices by changing both the magnetization and the electric polarization of the barrier layer. The techniques we use for growing these films, one of them is a physical technique referred to as pulse laser deposition. What we do here is to use a focused laser beam which hits a target, ceramic target in a vacuum chamber, leads to ablation of material from this ceramic target in the form of atoms and ions, which are transported to a heated substrate where under suitable conditions you can grow the complex oxide material. The other technique we use is chemical vapor deposition, where we use chemical precursors which are appropriately mixed in solution, and they are transported precisely using these mass flow controllers and then vaporized, and the solution then is transported to the oven where they decompose and grow the film on a substrate. And gases which are not decomposed, our precursors, are trapped and then pumped out in the exit. The second area involves oxide and chalcogenide nanostructures and films. We grow magnetic spinel oxides. Some of them are listed here. They are useful for a variety of biomedical applications. The second area is nanostructures and films for photocatalysis, primarily for water splitting, where we want to split water to hydrogen, oxygen for energy uses. So again, here we grow a variety of oxide materials or chalcogenide materials, which can be excited by solar radiation, and we can have plasmonic enhancement using metal nanoparticles which are embedded in these films to enhance the photocatalytic properties. The third area is solar cells, where we grow these complex chalcogenides, again as nanoparticles, which I talked about briefly earlier, and by changing the shape, size, and composition, we can vary the band gap, and these can be used as colloidal inks, so you can actually spray them on to large areas to make solar cell. The solution synthesis is very simple and straightforward with using appropriate precursors. We do chemistry, again, in a beaker or in a flask, which can be heated up. And under appropriate conditions, we can precisely grow nanoparticles, shown here, and their surface is covered with surfactants. On the right-hand side, you can see the transmission electron micrograph of these monodispersed nanoparticles, which are almost exactly the same shape and size for all of them. This shows you what we can do with them. So these magnetic particles can be dispersed either in an inorganic solvent, in an organic solvent, or in water. In this case, it is dispersed in organic solvent, it looks dark. By changing the chemical functionalization, we can have them transported in aqueous solution. And by bringing a magnet close to them, as you can see, they are attracted in both of them, both the cases, the solution becomes clear. And once we remove the magnet, they go back into suspension. So these are what are referred to as super paramagnetic nanoparticles. This shows you some micrographs, transmission electron micrographs of some of the complex chalcogenides I mentioned for solar rings. Again, by controlling the chemistry, we can change not only the composition, but the shape and the size of the spot. And thereby we can control their band gap. That means they show different color in solution, as shown here, in this case, by indium to gallium ratio. So we can have solar absorption over a broad wavelength range. Let me finally go to the bio-nano related research work we do. Here, the work is inspired by what nature does. Nature has perfected the growth of some inorganic materials, primarily calcium carbonide. Some of the examples are shown here, either nanostructures or layers. Here it is silicon oxide. Again, these are diatoms. There are a wide variety of nanostructures which nature produces. And here is an example where a magnetotactic bacteria actually synthesizes and grows precisely controlled magnetic iron oxide nanoparticles, which it then uses to navigate in salty water. Our intent is to expand this list of inorganic materials which nature has perfected to make other interesting nanostructures using biotemplates, primarily the proteins on the viruses, magnetic alloys, oxides, and chalcogenides. As you know, the viruses come in different shapes and sizes. These are examples of some different viruses. The one we have been primarily working on is this bacteriophage virus, which thrives on bacteria and is harmless to human being. It's a fairly large virus, and it's referred to as the P22 virus. What we do here is to genetically engineer the proteins either on the surface of the virus or on the inside, and thereby selectively nucleate and grow organic materials. Ultimately, the goal is to grow different organic materials in different parts of the viruses and thereby have a three-dimensional architecture. Here is an example where we have actually genetically engineered some of the surface proteins and thereby selectively nucleate and grow cadmium sulfide, which is a semiconductor material with a variety of applications. And by changing the amount of time, we can then grow the material all over the surface. Similarly, we can engineer the inside. In this case, nothing grows on the outside, but only in the inside. And as you can see, in this case, cadmium sulfide is mineralized in the inside. And again, by changing the amount of time and concentration, we can change the growth of cadmium sulfide. And we can readily monitor the growth using simple techniques like absorption spectroscopy, where we see the growth of the nanoparticles with time, and also monitor the disappearance of the precursors with time and control the band gap. So the ultimate goal is to, as I said, to grow three-dimensional architectures, either on the outside of the surface of the viruses or on the inside of the viruses. And we can play different tricks. For example, we can grow different inorganic materials on different parts of the outside of the surface, by genetically engineering them to selectively nucleate specific inorganic materials. The ultimate intent is to have a three-dimensional architecture, for example, something that will selectively absorb solar radiation, then transport the charge, and then convert them into electricity. So Hopefully, this gives you a brief overview of the work in my group. And if you are interested, feel free to contact me and I can provide you more details. Thank you very much for your attention and bye now.